Hello everyone, my name is Laura Sisk Hackworth and I'm in Dr. Kelly's lab. In Dr. Kelly's lab, we do a lot of studies on the microbiome. And for me specifically, I study the gut microbiome during puberty. Now, let's talk a little bit about what the gut microbiome is and why it's important. So, the gut microbiome is made of all the microbes in your intestine, and it's bacteria, viruses, maybe some fungi, and it's really important because the gut microbiome does a lot of different functions for the host that is important for our health and in some cases for disease development. So I'm really interested in the gut microbiome during puberty because a lot of gut microbiome related diseases arise during puberty. Some of these are polycystic ovarian syndrome, irritable bowel syndrome, and diabetes. Now, the reason that I think puberty is important is because we see sex-specific patterns in the gut microbiome in adulthood, but not in children. So in children, most gut microbiomes are going to look pretty much the same, regardless of whether they're male or female. But then during adolescence, we see a divergence of the gut microbiome into male and female patterns in adulthood. So the period I'm interested in is puberty here, when this divergence happens. And I'm interested in why this happens. So one way that I'm going to look at this is through a mouse study. And the mice I'm going to use are called HPG mice because they are lacking the gene that causes the hypogonadotrophic axis to start puberty. And so I'm going to have some mice where the male and the female go through puberty, just like normal. So pre-puberty, I'll expect to see pretty similar patterns in the gut microbiome, and post, I'll expect to see differences between male and female adult gut microbiomes, but in the HPG mice, which are different because they do not go through puberty, I'll expect pretty similar gut microbiomes before puberty, again, just like with the other mice, but then after puberty, I'm going to expect not very much divergence between the male and female adult gut microbiome patterns. So one of the reasons that I'm going to look at this is because in some preliminary data that I've looked at in just female mice, I've seen a difference between the pre and post gut microbiomes. And so those differences that I'm talking about are in two types of diversity that we typically look at with gut microbiomes, the alpha diversity and the beta diversity. Now, alpha diversity is about the number of species present. So if we have, say, 3 different species, which I'm going to show with shapes, in one group, and then in another group, maybe we have the same number of bacteria, but we only have 2 species. We've got squares and circles, and over here we've got a square, a triangle, and a circle. So that would be a difference in alpha diversity, 2 species versus 3. Whereas I also saw differences between pre and post puberty, not only in alpha diversity, but in beta diversity, which looks at differences between samples. So one way that you can look at this is so this would be 2 versus 3. Whereas if you look at these samples with beta diversity, you would see that this sample has 2 squares, and this sample has 1 square, and this sample has no triangle, and this sample does have a triangle. So that's looking at beta diversity. So between pre and post puberty mice, I saw differences in both alpha and beta diversity, which is why I'm going to look at puberty. So to conclude, I'd like to say that looking at puberty in the gut microbiome is really important for understanding how our gut microbiome develops for both health and disease reasons during puberty.